Good morning, everyone. Welcome to our last session of the week of our first Antibiotic Awareness Week lecture series. Today's presentation is going to be about newly approved antibiotics by Dr. Khalid bin Saleh, who is an infectious diseases clinical pharmacist and an assistant professor at the College of Pharmacy at King Saud bin Abdulaziz University for Health Sciences. He is my colleague here at National Guard, and he shares our infectious disease clinical responsibilities. So I'm really excited to introduce Dr. Khalid today. Dr. Khalid, please go ahead and start your presentation.
Okay, perfect. First, I would like to thank you, Dr. Anur, for the invitation and organizing this great, lecture series. And also, I would like to thank you all who attended this or who's attending this live seminar. So today, inshallah, my topic will be about newly approved antibiotic. But also, I will talk about the critical need for new antibiotic against bacteria, and specifically gram-negative bacteria. I will talk about also the newly approved antibacterial agent, including the doses and side effects. And before I talk about that, I will talk about the agents that we have in the preclinical as well as in the developing phases. And finally, I will talk about the newly approved antibacterial agent specifically against gram-negative microorganisms. Antibiotic resistance is a normal phenomenon. As we know, bacteria, fungus, as well as viruses are living organisms and they adapt to survive. However, right now we are seeing an accelerated pace in developing mechanisms of resistance. And actually, we have a lot of microorganisms now that are resistant to our big gun antibiotics, which can lead to microorganisms or infections that are extremely difficult to treat, which can lead to increased mortality, morbidity, as well as length of stay, and also cost expenditure in our health system. In the US, the CDC published a report that showed that at least or almost 3 million people are infected with antibiotic-resistant bacteria or fungi, and more than 35 people die as a result of these infections. And what's actually is interesting is that the CDC published a report in 2013, and then they published another report in 2019. And due to the effort they did to overcome microorganism resistant, we actually saw a significant decrease in mortality. And when I talk about the report in the next couple of slides, we will talk about the effort, what changed due to the effort made by the CDC. And to fight antibiotic resistance, as mentioned by my colleagues, it needs everyone's involvement. It needs a multidisciplinary team that can implement a good stewardship. And one of the ways to fight off antibiotic resistance is to have new antibiotics in our arsenal to overcome the resistant mechanism that we see on daily basis if you work in a hospital as the National Guard hospital. So this report from the CDC, as I mentioned, is the second of its kind. It presents data about the top 18 pathogens that requires our immediate attention. And compared to the 2013 report, I mentioned the mortality was lower in the 2019 versus the 2013. Also, the CDC reported an 18% reduction in overall death from antibiotic resistant. Also, a 28% reduction in hospital mortality due to antibiotic resistant. What's interesting too is that we had almost 30% reduction in Carbapenem resistant Acinetobacter, as well as Pseudomonas aeruginosa. But the bad news is that we had a 50% increase in ESBL. So the CDC divided the microorganisms into 3 categories: urgent, serious, and concerning threats. And the factors on putting microorganisms in these categories are clinical impact, economic impact if available, the incidence, the 10-year projection of the incidence, the new infections over the next 10 years, how easy a germ can spread or causes an infection, availability of effective antibiotics, and barriers to prevention. So the first microorganism I will talk about today is the Carbapenem-resistant Acinetobacter. As we know, Carbapenem-resistant Acinetobacter causes pneumonia, wound, bloodstream, and urinary tract infections. These infections tend to occur in patients in an intensive care unit. We can see that almost or more than 8,000 estimated cases in hospitals in patients in 2017. The mortality was 700, and the estimated healthcare cost was more than $250 million. And the problem also with Acinetobacter is that it can be resistant to nearly all antibiotics, and there are only few drugs that in development right now. And the medications we usually use for these microorganisms can be extremely toxic and can actually have poor concentration, usually in certain locations. The second microorganism is Carbapenem-resistant Enterobacteriaces. Also, this is an urgent threat level based on the CDC. It's estimated more than 13,000 cases in hospital with more than 1,000 mortality and more than $100 million in healthcare cost. Carbapenem-resistant Enterobacteriaces, they are a major concern for patients in healthcare facilities. Some bacteria actually in the family are resistant to nearly all antibiotics, also leaving more toxic or less effective treatment options. However, now with medications actually are getting approved, we have multiple options for these microorganisms that I will actually discuss when I talk about the medications approved antibiotics. Next is the ESBL, Enterobacteriaces. Almost 200,000 cases in hospital in 2017, more than 9,000 deaths in 2017, and more than 1 billion estimated healthcare cost. ESBL producing Enterobacteriaces are a concern in the healthcare setting as well as in the community. The problem is that about 1/4 of patients with these infections had no known underlying health conditions, and actually a lot of them were in the community setting. So we see in ESBLs in hospital acquired as well as in a community acquired. And also the antibiotic options to treat ESBLs are limited. And other than urinary tract infections, right now we have carbapenems to actually use it for a patient with ESBL. I know there is a debate when it comes to ESBL and other medications, but basically broad spectrum antibiotics actually used for ESBL producing microorganisms. And with multi-drug resistant Pseudomonas, this is a serious threat levels, more than 30,000 estimated cases in hospital, more than 2,500 estimated death in 2017, and more than $700 million in healthcare costs in 2017. Pseudomonas aeruginosa causes many types of healthcare associated infections. It can cause bloodstream infection, urinary tract infection, and a lot of other actually site infections. And we have a lot of cases where Pseudomonas aeruginosa, they are pan-resistant to our antibiotics. So having new medications or approved medications to fight these kind of infections that has actually a novel mechanism of action is really important and recommended. And I will talk about the CDC report actually with the global priority list of antibiotic resistant bacteria to guide the research, discovery, and development of new antibiotics. And actually today, my talk is going to focus about gram-negative microorganisms that affect usually patients worldwide. So with the shedding light to these resistant microorganisms, especially when we talked about the reports that came out from the CDC, in 2017, the WHO published its first ever list of antibiotic resistant priority pathogen. This list was drawn actually to guide and promote research and development of new antibiotic And as part of the WHO's effort to address, as we can see, actually a growing global resistance to our current antimicrobial agent. So the WHO list is divided into 3 categories according to the urgency of the new antibiotic: critical, high, and medium. So you can see the list here for critical need for antibiotics against gram-negative microorganisms, including microorganisms that we saw in the CDC report that imposes great risk for patients worldwide. We can see that Carbapenem resistant Acinetobacter, we don't have a lot of good options when it comes to treating these microorganisms. Carbapenem resistant Pseudomonas, as well as Carbapenem resistant Herobacteriaces and a 3rd generation cephalosporin resistant or can be ESBL producers. And for the less critical need for antibiotics, or the high need for antibiotic, is with we can see that mainly our gram-positive microorganisms with the Enterococcus faecium vancomycin resistant, or methicillin resistant vancomycin intermediate and resistant Staphylococcus aureus. And you can see the other actually microorganisms that the WHO recommended to guide the new approval or development of antibiotics to actually fight these type of microorganisms. So, and part of the effort from the WHO is actually to encourage governments to put policies and incentives for drug companies for developing antibacterial agent. For example, we saw the FDA actually encouraging drug companies with extending their patency or accelerating the approval of the antibiotics in general. And the reason for that, you can see actually the timeline for classes or new classes of antibiotics approved. Since the 1920s, where we actually saw penicillins, until the 1987, the lipopeptides. After that, there was no new classes of antibacterial agents were developed until recently. And due to the drug resistance that we are facing right now, this can be a huge problem. And even with the newly actually approved new classes of antibacterial agent that we actually saw recently, they do not address the need for the multi-drug resistant microorganisms, and most of them, they are the place in therapy is still not defined, and definitely we're not going to see it used if we talk about our hospital, or for really severe multi-drug resistant microorganisms. So what happens is the WHO, they are publishing documents about the new antibiotics in the preclinical and the development phases to actually to try to expedite the approval or the development of microorganisms that can actually meet the need for the priority list they published. And for the new antibiotics in the preclinical phase, before the phase 1, phase 2, phase 3 trials, usually antibiotics will go to lead optimization when the lead compound is undergoing multiple in vitro or in vivo screens to generate a very good pharmacological compound that has safety and pharmacokinetic profile that is appropriate. And then it goes to the preclinical candidate, which the lead compound that passes the initial toxicology test and demonstrates sufficient safety profile, which when combined with a suitable pharmacological efficacy, it will go to the CTA-IND phase, which they will study these compounds, including the absorption, distribution, metabolism, and excretion, before obtaining permission to begin human clinical testing. So currently we have 252 preclinical compounds under study. 38 products target the WHO priority list, and there is a significant number of products, almost 100 products, were focused on a single pathogen species. which is actually represent a shift toward pathogen-focused therapies rather than the broad-spectrum antibiotics that we usually use empirically for a patient coming without knowing the microorganisms, which is very interesting, knowing that most of the newly antibiotics or the antibiotics we use, usually they are broad-spectrum in nature, but having a single pathogen targeted product can be very actually appropriate or good when it comes to resistant and when it comes to antimicrobial stewardship specifically. And we can see actually that more than 50% of the single pathogen target product are against Mycobacterium tuberculosis and Pseudomonas aeruginosa. However, we don't know yet how many of these 252 will be actually approved and used in practice and actually be appropriate or something that can help us in fighting multi-drug resistant microorganisms. So for the new antibiotic in development phase, we have only 42 antibiotics right now in the clinical development, 15 in phase 1, 12 in phase 2, and 11 in phase 3. Unfortunately, there is a historical data that tells us that 1 in 5 agents that enter phase 1 trial will receive FDA approval, which is extremely low, knowing the need right now for new antibiotics to fight off the infections that we're facing on a daily basis. So since we're talking about gram-negative microorganisms, for antibiotic in development phase, only 16 agents currently tested against gram-negative microorganisms. And if the historic data are correct, then we might see only 3 approved medications for these gram-negative pathogens. And even after approval, we wouldn't know the effectiveness and the safety of these antibiotics against the multi-drug resistant microorganisms that we are really needing for a new antibiotic for. And when we talked about the priority list for WHO threat pathogens, only 11 of these antibiotics in the development have the potential to treat these pathogens. So now that we talked about the antibiotics and the pre-development as well as the development phase, we will talk about recently approved antibiotic that have the potential to treat multi-drug resistant gram-negative microorganisms. And the first medication I will talk about today is Svidricol. It was FDA approved in 2019. This is a novel combination of Sidrophor and cephalosporin antibiotics. So the uniqueness of this antibiotic is that it utilizes the iron starvation of a microorganism to actually penetrate the outer membrane. As microorganisms release a molecule called sidrophores that's actually scavenge for free ferric iron, Svidricol takes the chance to actually enter and penetrate the cell wall, which can lead actually to better when it comes to fighting infections that has multiple resistant mechanisms. And we'll talk about the indications or the place in therapy for Svidricol in the next few slides. So for Svidricol dosing, this medication can be removed from the kidney, so dose adjustment is necessary based on creatine clearance. The creatine clearance is based on Cockcroft equations. And what's interesting with this medication is that there was a pharmacokinetic study with this medication, and it found that with patients that have really good kidney function, more than 120, it showed less concentration. The usual dose for this medication is 2 grams every 8 hours, but after seeing that has low concentrations, actually they recommended to have the frequency into every 6 hours. So the side effects for this medication doesn't have severe side effects. Usually these side effects, what we see in beta-lactams in general, where the hypersensitivity reaction, which it's actually when it comes to structure, it can be similar to other cephalosporins. So patients who have allergy to cephalosporin for other cephalosporins, it can have a cross allergy with Svidricol. The most common side effect when it comes to this medication was GI side effects, We saw liver function elevation, hypokalemia as well. So the first study I will talk about today is phase 2 randomized trial, was published in 2018. It's Cefidricol versus imipenem celastin for treatment of complicated urinary tract infection caused by gram-negative microorganisms. So this is a multi-center, double-blind, parallel group, randomized, non-inferiority study. They compared Cefidricol 2 gram every 8 hours to imipenem celastin 1 gram every 8 hours as well, in patients with complicated urinary tract infection with or without pyelonephritis. So that included 371 patients, 252 patients in the Cefidricol group, and 119 patients in the imipenem celastin group. So they included patients who are adult, more than or equal 18 years old, with complicated urinary tract infections, and they excluded patients with a baseline urine culture of more than 2 pathogens, as well as pathogens known to be carbapenem resistant. The primary outcome was a composite of clinical response as well as microbiological response at test of cure assessment. It was defined as 7 days after the end of antibiotic treatment. And the secondary outcomes, it was microbiological and clinical response per specific pathogens. So about for the baseline characteristics, almost 60 to 40% female to male ratio in the 2 groups. The average age was 61 years old, and almost 50% of the patient had pyelonephritis. Out of these patients, the total patient, 100 patient had an ESBL organisms, 70 in the Cefidricol group versus 30 in the imipenem group. And you can see actually the distribution of the pathogen isolated at baseline. The most common pathogen in both group was E. coli, mainly almost 60% in both group. Next is Klebsiella pneumonia, which actually both of them can represent almost 80% of all microorganisms actually isolated from urine culture. So this study showed that Cefidricol was not inferior to imipenem celastin, with the primary outcome actually achieved. We can see that the composite outcomes was seen in 73% of the patient versus 55% of the patient. Specifically, the microbiological responses and the clinical responses was 73 versus 56, 90% for the clinical responses versus 87. So basically, this study showed that Cefidricol was not inferior to imipenem celastin, and when it comes to specific microorganisms, also was not inferior, almost 50% in both groups when it comes to primary outcome against Pseudomonas aeruginosa, and for ESBL producers microorganisms, it was 63% for the primary outcome versus 47%. So they did a post hoc analysis that actually demonstrated superiority of Cefidricol for the primary endpoint, the composite outcome, which basically was driven by the higher microbiological eradication rate. So what's the limitation of this study? The first limitation was the sample size definitely was small, and actually after this study and the preliminary data from other study, the phase 3 study actually granted FDA approval. And the main actually limitation for this study is that we want to test Cefidricol in multi-drug resistant microorganisms and carbapenem resistant microorganisms, other actually locations for bloodstream infection, pneumonias, and actually more severe infections in actually places where we actually we're going to worry about not urinary tract infection. But as we know, usually drug companies, especially their first studies, so they can grant the approval, they usually test patients for gram-negative microorganisms, usually in urinary tract infections. For gram-positive microorganisms, usually skin and soft tissue infections, because it's usually easier to treat, and basically it's not going to show any it's not going to be inferior to the compared antibiotics, which can lead to approval or FDA approval. So right now, we actually have multiple studies. We are waiting for the result and to be published for Cefidricol. There is a study to treat nosocomial pneumonia caused by gram-negative pathogens for best available therapy for treatment of gram-negative bloodstream infections. And what's really interesting is the study for best available therapy compared to Cefidricol for severe infection caused by Carbapenem resistant gram-negative pathogens, the Credible trial. And we're actually going to go back in the next few slides and talk about this study. So what makes this medication unique actually is the spectrum of activity. It has great activity against both lactose fermenting and non-lactose fermenting gram-negative pathogens. However, it has weak activity against aerobic gram-positives and anaerobic gram-negative microorganisms. So the placent therapy, the proposed placent therapy, it's actually showed in vitro trials showed great activity against Enterobacteriaces, Pseudomonas aeruginosa, and Acinetobacter. This medication is stable against all amplar classes, including class B, actually, metallo-beta-lactamases. So there are a lot of indications, and actually once it's approved or once it's actually used, it can have a lot of indications and can be great medication to be used for specifically our patient population. However, there is a bad news. So the bad news is that the FDA, they published a briefing. It was a meeting of the Antimicrobial Drug Advisory Committee about Cefidricol injection. And the problem with that is that when after the medication was approved, the drug company actually they showed the FDA or they released the result for their Credible trial all-cause mortality. So the Credible trial, it's not published yet, but the result was actually shared with the FDA. They compared Cefidricol to the best available treatment for Carbapenem-resistant Enterobacteriaces. Actually led to higher all-cause mortality from the beginning until day 49 when they compared Cefidricol versus best available therapy. We can see at day 14 mortality was 18% versus 12%. Day 28, 24% versus 18%, and day 49 was 33.7% versus 20%. The problem is that the FDA, when they released the document, they couldn't find justification for the increase in all-cause mortality. And actually, I was really intrigued, and I really wanted to know what's the reason for that. And I went back to multiple in vitro studies, and I found an in vitro study in intrapulmonary pharmacokinetic, actually, of Cefidricol. It was actually evaluated in phase 1 trial. Specifically in the alveolar macrophages, it showed low concentration. So whether that is going to affect patients who have pneumonia, whether it's a hospital-acquired or ventilator-associated pneumonia, it can affect the outcome for those patients. This is actually what we really need to know. But what's interesting is that in the trial, the majority of patients had baseline diagnosis of pneumonia. Actually, 44% of the Cefidricol group and almost 45% in the best available therapy, and of which almost 42% of those patients actually died in the Cefidricol group versus 18% in the best available treatment. However, we really cannot really say that this is the reason for the higher all-cause mortality, and we wouldn't know until all the published, all the studies that are undergoing right now, which one of them is for pneumonia, which I believe it's already they finished actually the study, and we're waiting for the published article. So until we actually see the data, we will know for sure what's the reason for the all-cause mortality. But this medication can be a great option for all the microorganisms we're facing, specifically the gram-negative microorganisms, and hopefully with the published data comes out, we will know for sure what's the place of therapy for these microorganisms or antibiotics. So, and the next medication that I will talk about is Meropenem Vebromer. This medication was approved in 2017. The mechanism of action, obviously we know about meropenem. This medication we've been using for a long time. It inhibits cell wall synthesis by inhibiting penicillin binding protein, causing cell death. And the Vebrobactam, this is a new beta-lactamases inhibitors. It prevents beta-lactamases such as KBCs from hydrolyzing meropenem. So this can be a really good option for patients with carbapenem resistant Klebsiella pneumoniae and other actually microorganisms that we'll talk about in the next few slides. With Meropenem Vebromer, you can see the dosing for this medication. Also, it can be removed from the kidneys, so dose adjustment is required for this medication. And side effect is not going to be different from meropenem, the hypersensitivity reaction we usually see with the beta-lactams. CNS side effects, specifically seizures in patients with history of seizure, where it actually decreases that seizure threshold. So all these can be seen with the Meropenem Vebrobactam. So the first study that we'll talk about is a very famous study, the Tango-1 randomized clinical trial. This is a phase 3 randomized trial, was published in 2018. It's the effect of Meropenem Vebrobactam versus piperacillin tazobactam in clinical cure or improvement and microbial eradication in complicated urinary tract infection. Again, usually the first studies or for any new medications, new medication to be again for urinary tract infection. So this is a multi-center randomized double-blind, double-dummy active control non-inferiority trial. They compared Meropenem Vebrobactam, you can see the dose, to 4 gram total over 3 hours to piperacillin tazobactam. So they included patients who are adult with complicated urinary tract infection, including acute pyelonephritis, and they excluded patients with creatine clearance less than 30. So the primary outcome, they had the FDA criteria as well as the European Medicine Agency criteria. The FDA criteria is a composite outcome of clinical cure as well as a microbial eradication. And for the European Medicine Agency criteria is the microbial eradication at the test of cure visit. The baseline characteristics was actually similar in both groups. 60% of the patient had acute pyelonephritis and 40% had complicated urinary tract infections. Same thing when it comes to urinary tract infections, but we usually see is E. coli and Klebsiella pneumonia. And it's the same thing, almost 80% in both groups actually having these two microorganisms. So this study proved that Meropenem Vebrobactam was not inferior to piperacillin tazobactam when it comes to the FDA criteria as well as the European criteria. 98% versus 94% for the FDA criteria and 66% versus 57% in the European criteria. So the problem with this study, almost all pathogens were susceptible to meropenem, but about 12% were resistant to piperacillin tazobactam. However, they removed the patient who were resistant to piperacillin tazobactam, and they did actually they repeated the statistical analysis and was non-inferior piperacillin tazobactam or meropenem non-inferior to piperacillin tazobactam, even with removal of those 12% of patients that are resistant to piperacillin tazobactam. Also, this trial was not designed to evaluate carbapenem-resistant pathogens. The real indications for these microorganisms is for the severe infections. serious other infections in different places other than the urinary tract infections and in the really multi-drug resistant microorganisms. So in this trial, it wasn't assessed. They only assessed patients with urinary tract infection or pyelonephritis, which is usually easy to treat, and not a lot of resistant or multi-resistant pathogens. And then actually comes out the Tango 2 trial. It's a phase 3 randomized trial. It was published later that year to assess the efficacy and safety of Meropenem Vebromer versus best available therapy in patients with Carbapenem-resistant Enterobacteriaces. So this randomized, prospective, multi-center, multi-nation, open-label, active control trial, they compared Meropenem Vebromer 32 patient to best available therapy, 15 patient, an adult with infections due to confirmed or suspected CRAs, Carbapenem-resistant Herobacteriaces. So what's included in the study as a best available therapy was carbapenems, tigacycline, colistin, polymyxins. It can be a combination of all these medications, also aminoglycosides, or Saftasidim avibactam as a monotherapy only. So they included patients with suspected or confirmed CRE infections. Infection type, they included bacteremia, urinary tract infections, acute pyelonephritis, also hospital-acquired pneumonia and ventilator-acquired pneumonia, as well as complicated intra-abdominal infection. So as we know, with Meropenem Vebromer, they are actually not stable against NDMs, the metallo-beta lactamases, so they excluded patients who are that's Carbapenem-resistant Herobacteriaces that producing New Delhi metallo-beta lactamases. Also, they excluded patients on CRRT. So the primary outcome was clinical cure at test of cure and end of therapy, microbiological cure at test of cure and end of therapy, as well as 20 days mortality. The baseline characteristics, Klebsiella pneumoniae was the most common pathogen. Bacteremia was identified the most common infection, followed by urinary tract infection and acute pyelonephritis. Also, what's interesting that 9 patients with prior antibiotic failure were included in the Meropenem Vebromer group. So the result for the primary endpoints, the clinical cure at end of therapy, as well as test of cure, microbiological cure, as well as for 20 days mortality, it was actually non-inferior to the best available therapy. You can see with the clinical cure 65% versus 33%, microbiological cure 65% versus 40%. 28 days mortality was 15% versus 33%. Actually, this study is was descriptive, and there was no formal power or sample size calculation was performed. So it's really difficult to actually interpret the results as significant or not. And actually, when I talked about the patient who had a previous clinical failure, when those patients were excluded from the 28 days mortality analysis, only 1 patient in the Meropenem Vebromer group actually died versus 5 patients in the best available therapy. Again, the sample size is actually small, so it's extremely difficult to actually think this as a clinical significant until we have a better and bigger actually study. So the limitation, as I mentioned, the small sample size, study was descriptive, no formal power or sample size calculation was performed. People had a variety of infection sites were included, so it's really difficult to interpret the result for a specific infection or a specific location of infection. So with Meropenem Vebromer, we have actually a very good medications that recently approved. It can have a lot of actually indications especially when we think about our patient population that have multiple drug-resistant, carbapenem-resistant Herobacteriaces. So this medication can have actually a great role in fighting off multi-drug-resistant microorganisms that we usually see on a daily basis. Last medication I will talk about that this is something we have in our formulary, Saftasidim avibactam. It was approved in 2015. We all know Saftasidim. This is a third-generation cephalosporins. It inhibits cell wall synthesis. It binds to the penicillin binding proteins. Avibactam, this is a first-in-class non-beta-lactam beta-lactamases inhibitor. So Saftasidim avibactam, also a medication, can be removed from the kidney. So you're going to see dose adjustments based on kidney function. Also, when it comes to side effects, it's what we usually see with Saftasidim, hypersensitivity reactions. It can cause elevation in the liver function tests, and all these side effects we usually see with cephalosporins. So with Saftasidim, it has actually really great role when it comes to treating patients with multi-drug resistant microorganisms. So usually the place in therapy, there are multiple in vitro studies that showed these medications that's stable against ESBLs, MC producers, carbapenem resistant Klebsiella pneumoniae, carbapenemases, OXA48 producing Enterobacteriaces, also drug resistant Pseudomonas aeruginosa. So it can have actually, and we actually use it often for our patient population when they have really multiple drug resistant, and there is not a lot of good option to actually fight these infections. Keep in mind with these medications, it has limited activity against gram-positive microorganisms, because we know actually Saftasidim doesn't have really good gram-positive coverage. Acinetobacter and Stenotrophomonas in vitro studies showed higher MICs or elevated MICs. So it can be actually, or maybe we should think about before we use this medication for these microorganisms. Also for ampler class B enzymes, specifically metallo-beta-lactamases. However, I will talk about what if we added azithromycin for those patient population with this type of infection. So the first study that I will briefly talk about is the Reprove Trial. This is a randomized, double-blind, non-inferiority trial, assess the efficacy of Saftasidim in treatment with hospital-acquired pneumonia, including patients with ventilator-associated pneumonia. They compared Saftasidim to Meropenem. So they included around 880 patients, and the primary endpoint was clinical cure rate at the time of cure. And you can see actually in this graph, Saftasidim was not inferior to Meropenem in treatment of adult patients with hospital-acquired pneumonia, including ventilator-associated pneumonia. And when actually it was specified for a VAB without HAB, also it showed non-inferiority for Saftasidim versus Meropenem. And what's also interesting is that they compared patients who had Saftasidim susceptible and Saftasidim non-susceptible to Meropenem. And also for those patients, it was non-inferior to the Meropenem group. And also they assessed the all-cause mortality at day 28 in the Saftasidim group. Meropenem group was actually, Saftasidim was 8.4% versus 7.3%, and I was actually non-inferior when it comes to 28 days all-cause mortality. And in Recapture trial, it was Saftasidim versus Doripenem for treatment of complicated urinary tract infections. They include acute pyelonephritis. You can see that mainly they focus on if it's Saftasidim non-susceptible versus Saftasidim susceptible pathogens, and also they had specific microorganisms and if there is actually any different when it comes to efficacy for Saftasidim versus Doripenem. And this study also showed actually reached the non-inferiority when it comes to Saftasidim compared to Doripenem when it comes to all pathogens. or specific pathogens, and also when it's non-Saftasidim susceptible pathogens, as well as Saftasidim susceptible pathogens. So as I mentioned earlier, what if we added azithromycin in patient with metallo-beta-lactamases? So as we know, azithromycin is stable against metallo-beta-lactamases. Activity usually lost against these organisms because that it will co-produce ESBLs or MC enzymes, and usually or often actually they produce other mechanism of resistance such as ESBLs or MC, which usually azithromycin is not stable against these enzymes. So clinical experience as a monotherapy for azithromycin is lacking for MBL producers, and it's actually not recommended to use azithromycin by itself, because as we know and mentioned just now, is that usually with metallo-beta-lactamases there is other mechanism of resistance. There was some in vitro data support the use of a combination of azithromycin with Saftaz-AVI, but there is not a lot of clinical studies. However, there was a recent study was published in 2020, was actually May 2020. I wasn't able to find the entire study, so I was actually I only looked at the abstract, just to be honest, because this was a very interesting study. It was comparing patient with the Saftaz-AVI azithromycin versus best available therapy in patient with bloodstream infection due to metallo-beta-lactamases. And actually the primary endpoint was 30-day all-cause mortality. Most of the microorganisms were New Delhi strains, Klebsiella pneumoniae, and the study found actually the 30 days mortality rate was almost 19% in the Saftaz-AVI azithromycin group, versus 44% in the best available therapy. So this is actually an option that can be, we can actually think about when a patient have metallo-beta-lactamases. And just to actually emphasize more on those two combinations, Saftasidim doesn't really have a role in fighting metallo-beta-lactamases. So what we really want from that combination is the azithromycin and the avibactam. And currently there is a medication that's in, I believe it's in phase 3 trial. There are multiple studies are undergoing for azithromycin and avibactam for metallo-beta-lactamases. So once this combination actually is approved, we can have a good actually medication for metallo-beta-lactamases producing microorganisms. Right now, we can actually use these combination for a patient with metallo-beta-lactamases. So just to conclude, my conclusion, as we know, antibiotic-resistant infections, they are global issue, affect patients worldwide. The WHO emphasized on the need for no antibiotics for critical microorganisms. Unfortunately, the success rate for the clinical drug development is low, and also the medications that under actually development right now, not a good percentage actually meet the need for the critical microorganisms for our patient population. There are multiple antibacterial agents are approved for gram-positive and gram-negative microorganisms. We have a lot of actually good antibiotics that is approved for our multi-drug resistant microorganisms. More data is needed to actually real assess the place of therapy for these antibiotics. Thank you all for listening to my talk, and I'll take questions.
All right, excellent. Thank you very much, Dr. Khalid. That was very thorough, very informative about the new medications, and it's a really nice way to end the week after talking about all the problems that we're having with antimicrobial resistance, and then ending it with some solutions and some hope for some of our really resistant bugs. So thank you for that. And I guess I'm really excited to see, especially for cefiderocol, what we could use, how we could use it in our patients with especially Acinetobacter MDR. So, inshallah, looking forward to that. Um, Within your review, did I mean, it seems like still, even though there's quite a bit of research being done, it seems that we still need, we have a long way to go to get to more useful antibiotics for our area. Um, what would you say would be, as a summary for our audience now, um, like if you can categorize the medications that are available with the possible options that they could treat. So let's say you mentioned at the end Saftaz Avi with Astrienam for NDM. What about Oxa 48, for example, or yeah?
Yeah, thank you. This is a great question. Thank you, Dr. So actually, we have a couple of options we have right now for OXA 48. So now we're using definitely combination therapy, but there are a lot of new agents that can be actually used for specifically OXA 48. For example, when we talked about the Meropenem Vebromer, there are multiple in vitro studies that showed actually stable for these microorganisms. Sofidricol is an option for these microorganisms as well. Ceftaz-Avi is something we can actually use. So we have multiple new agent we're using right now for these microorganisms. And when you talked about Acinetobacter, the problem is there are multiple or in vitro study with Sofidricol. It showed actually it started with actually MIC that is appropriate or low MICs, but it had increased after introducing Sofidricol in vitro for Acinetobacter. So this is something we can actually worry about when it comes to using Sofidricol in patients with Acinetobacter. And when I went back to the study that had higher, the credible trial that had higher mortality, a lot of patients who had Acinetobacter actually had higher mortality than the other microorganisms. This is something we should worry about and something we should think about, especially that we don't have the full article, the full data about these about Sofidricol and the reason for the higher mortality. But yeah, thank you for the question, Dr.
Yeah, exactly. Thank you for summarizing that. That's just kind of what I was alluding to. So it sounds good. But maybe just for the audience, I just thought about one thing. It's important to also realize, like, I know we're talking about a lot of technical things, but when we talk about NDM and OXA 48, it's the same as what you, or can you describe, how would you easily describe what OXA 48 and NDM is for our audience, or, as opposed to KPC and IMP? What are they exactly?
So they're basically a mechanism of resistance. So it's how the microorganisms actually fight the antibiotics. And this is basically what we actually think about when we think about the mechanism of resistance.
Yeah, exactly. Yeah,
Yeah.
that's exactly it. Like, so I know, because we're using acronyms and it may be a bit confusing if someone is not, as involved as we are in the ID issues. But so basically, just for some of the audience that may not be involved. So when we talk about OXA 48, this is an enzyme that is actually, it's not an enzyme. It's a gene responsible for the production of an enzyme that can break down an antibiotic. So when we say, and what do you think, Dr. Khaled? So when we say, for example, that a bug has NDM, so Nucleoli beta-lactamase, so we expect it to produce an enzyme that can that is stable against, for example, Meropenem. Would you say, or how would you describe it?
So NDM is stable against
Meropenem, for example. So what I'm trying to allude to is, because there's a question here in the audience which asks, what's the advantage of Zerbaxa over Avicaz? So I'm trying to segue to that. So maybe and I don't like to use brand names, maybe generic names. So Zerbaxa is ceftolozane tazobactam over Avicaz, which is Saftasidim avibactam. So what would you say is the advantage of one over the other?
If we think about actually ceftolozane, it had better advantage when it comes to Pseudomonas aeruginosa. There are multiple in vitro studies that showed actually better MICs for ceftolozane than the Saftaz-Avi. And for Saftasidim avibactam, it actually had better when it comes to gram-negative Enterobacteriaces that Carbapenem resistant. So basically what we usually think about when we think about Saftaz-Avi is better when it comes to Enterobacteriaces that are Carbapenem resistant, and for ceftolozane, it's usually for Pseudomonas aeruginosa that is multi-drug resistant.
Yes, exactly. And the reason for that is because Enterobacteriaceae are more likely to be OXA48 holders rather than Pseudomonas, which has a different mechanism of resistance, and that's why ceftolozane tazobactam can be more stable with Pseudomonas, basically. So I just wanted to point that out. Otherwise, would you? We have no other questions. Would you like to add anything, as you know, for our employees here at National Guard? Would you like them to know anything specific about our available agents?
Oh yeah, actually, right now we have Saftaz-Avi and ceftolozane in our formulary. We should definitely utilize really good antimicrobial stewardship, because we don't want to lose those two medications as well, because if we start using them when they are not needed, we're gonna definitely lose those medications. The thing that I really wanted, if you are interested in knowing the antibiotics that right now being tested for different microorganisms, there is a website, the pewtrust.org. They actually, every 6 months almost, they publish, this is actually a it's a charitable organization. They are really interested in multi-drug resistant microorganisms, and they have list of all the medications, the antibiotics that they are in development currently. They have their what phases they are right now, and what's the coverage when it comes. Does it cover MRSAs? Does it cover Enterobacteriaces? And what's the specific genes that usually they target? So all these are available if you are interested in pewtrust.org. You can go to the list of the antibiotics, and then in development, actually, if you are interested, definitely.
Yeah, that's a great idea. That's absolutely, the Pew Trust is really interesting to go through that website. All right, so thank you very much. This was a very, very interesting presentation, and I hope our audience enjoyed it. If you have any more questions, please feel free to reach out to Dr. Khalid, and this is his email. So thank you all for attending. I hope you enjoyed the Antibiotic Awareness Week activities this year. For our National Guard and non-National Guard audience, thank you for joining us, and we'll see you next year, I hope.
Thank you. Thank you all.